Hey cell biology students, this is Dr. Martin coming to you on a Tuesday morning. It's kind of brisk, kind of cold out here. That's why I have my stocking cap on this bald head. It's because I'm cold. But I'm coming to you from the Rogers State University Scientific Research Station. It's about a 270-acre research station that we use for our environmental conservation degree program in biology. We do research here, we do laboratories here, and it's a pretty cool place, even in the wintertime on a cold morning. But it's a beautiful blue sky, the sun's out, and a little bit windy. But hey, I wanted to talk to you a little bit about this week and the final week of classes. We're covering two chapters, chapter 16 and chapter 17. I've already started chapter 16, where we covered and reviewed the structure of the DNA molecule a little bit more in depth than we did earlier in the semester. And we also talked about the most important thing, and that is the replication of the DNA molecule. That's where in S phase, when a cell reaches a certain volume and decides to replicate itself from parent to daughter cells, making sure we have the exact genetic information in those daughter cells that we had in the nucleus of that parent cell. And that occurs in S phase. So every single chromosome has to replicate itself. Exactly. An amazing statistic in your book in chapter 16 says that if we counted every single nitrogen base, sequence of nitrogen bases, C, G, A, and T, out of the billions of nitrogen bases in the human genome, if we wrote them in 12 font, it would include about 14 different biology texts. That's how much information is in one nucleus genome of your body. It has to be replicated exactly. We talked about the enzymes that are involved in that replication process. Helicase, DNA polymerase, and DNA ligase. Those are the three primary enzymes involved in that replication process. That's chapter 16. Chapter 17 is different, a similar process, but a different process. This is where a cell uses the information on just one small segment of a chromosome. Chromosome 1, 3, 14, really doesn't matter. One small segment of nitrogen bases, a precise sequence of nitrogen bases. How does it use that gene to make things inside of your cells? Generally proteins, mostly proteins. How does it know to make insulin? How does it know where to go get that sequence of bases to build insulin? Or collagen, or hair follicles, or other proteins in your body? That's what we call protein synthesis. That's chapter 17. That's where one small segment of a gene separates by an enzyme, breaking those bonds in between the nitrogen bases. And then RNA, ribonucleic acid, nucleotides in the nucleus, they bond to those nitrogen bases in the 5 to 3 prime or 5 to 3 carbon strand or direction. It reads that one sequence, starting with the start, ending with a stop, and then it releases in a messenger RNA strand, migrates out of the nucleus through the nuclear membrane to a ribosome in the cytoplasm. How does it do that? And then the DNA molecule just glues back together, because DNA doesn't migrate in and out of the nucleus. RNA does. This messenger RNA string now goes out to the cytoplasm, attaches to a ribosome. Another type of RNA called transfer RNA has a sequence of 3 nitrogen bases on the end of the molecule. It's built kind of like a cross. And at the bottom end of this molecule are 3 nitrogen bases. It corresponds, certain nucleotides will correspond with the first 3 nitrogen bases on that messenger RNA strand. On that messenger RNA, those first 3 nitrogen bases are called a codon. On the transfer RNA molecule that's across, the 3 nitrogen bases that correspond to it are called the anticodon. They match up. On the other end of that molecule is an amino acid. The next transfer RNA molecule matches up, anticodon to codon. On the other end is an amino acid. Those 2 amino acids bond. A third transfer RNA molecule, anticodon, bonds with the codon. Another amino acid, So a long string of amino acids are now being built, which is a protein. The anticodons bond to codons on that messenger RNA. The code is exact, and it matches exactly to that DNA strand that was in the nucleus. That's how your body, that's how your cells build things. That process in the nucleus, where we transcribe the sequence of nitrogen bases, is called transcription. The process out into the cytoplasm, where we match up codons with anticodons, is called translation. That's what we're going to talk about in chapter 17. The notes are already up in MyRSU for both chapter 16 and 17. Remember, there's a little link on each slide where you can click and you get to hear my wonderful voice that I recorded in my nice warm office. I will lecture on every slide. There's also a few review questions for chapters 16 and 17, and they may be combined. I know the quiz is combined. There's some quiz questions that are combined for those two chapters, and then there's review questions for both of those chapters. I didn't put any readings up in chapter 17 out of your chapter because it goes so much more in depth than what we're talking about. But this is chapter 16 and 17. The exam will be next Tuesday morning. It will start at 8:30, or really any time between 8:30 and 10:30. You have about 50 minutes to take a 58 question test starting next Tuesday morning at 8:30. You don't have to start at 8:30, but you have to be done by 10:30. So leave yourself 50 good minutes to take that exam. 58 questions, 50 minutes. It's all in MyRSU. There will be a link there for exam number 5. That's what you click on. So exam number 5, next Tuesday morning, chapter 16, chapter 17 only. Hey, I might send another video late in the week or maybe next Monday just to make sure we're all on the same page. Give me a call or send me an email if you have any questions. See you later.